Okay, we have heard a lot about the need of reference databases, and we were lucky enough to get some funding for doing that for the marine domain to the Accelerate project. So I want to tell you a little bit about these databases and how we actually made those. So a little bit about the background. It was a need for more specific marine metagenomics databases or resources. And of course, the main was actually to make new reference databases. What we want to do was to include sequence and metadata, the contextual data that is describing the sequences, from sampling to throughout the whole process until archiving, more or less. We will follow the FAIR principles, and that is findable, accessible, interoperable, and reusable. So everybody should have access to it, and of course can reuse the data in a simple way. We should provide the metadata of the highest possible quality, and that often means manually curation. So you have to sit down, go into the literature, and find the metadata, because it's not there. We should also, of course, link to other important resources, and that is mainly the major databases that is out there. And we should use existing standards and best practices. So that was more or less the background for starting the project. So we ended up with not only one, but actually 3 different databases. What we call the Marine Reference Database, the MAREF, and that is more or less not a gold standard, but probably a silver standard. So that should contain only complete genomes. The MARDB, the aim was actually to include the whole genome shotgun, the single-cell genomes, and the metagenome assembled genomes, because they are often not complete, but still it has a lot of values. And the last one was the Marine Microbial Gene Catalog, the MARCAT, and that contains all metagenomic projects, and the sequences should be obtained by using MetaPipe for predicting the genes and the annotation. So we set up a plan for how to do it. Of course, we have to start with the beginning. We need to collect the data. We need a curation step. We need to refine everything. We have to translate it from a human readable to machine readable language. So from text separated columns to XML files. And of course, we have to make the databases. And we need to collect the sequences or the genomes from ENA. From the genomes, we just use what is inside already, but the metagenomes, we have to run it through MetaPipe, and then we have the BLAST databases, and we have a link between those. And that is accessible through the Marine Metagenomics portal. So to go into more detail, so this is the databases that we used to collect the data. So we used, of course, the sequence databases. We used contextual databases, taxonomy, bacterial diversity metadatabases, culture collection. It's a lot of information in these databases. And of course, the marine organism database. We did some mapping of the location where the sample was actually sampled. Literature, PubMed, Europe, PMC. We have to follow the standards for genomes and metagenomes. And of course, we used ontologies. And that is a defined set of describing environments, for example. So that was the resources that we started with. And of course, we had to make a curation database. So this is made in in Base. So this is all the metadata, that we have put into the database. In total, there are 108 different metadata fields. And this has been manually curated. So you can understand that this is a huge, huge task to sit down, go through every genome and to find the metadata, because often it's not described. So you have to go into the literature and find the data. So you can see we have, of course, a summary of the data. We have information about the organism and taxonomy, how it was isolated, if it is associated with a host or other kind of information, phenotype type information, annotation information, assembly. All of the data should be there. That is available either through the sequence files or through literature files. So it is often a big problem. This is just an example of one of the columns. If you're taking, for example, the depth, you can see it varies. So somebody is just saying 10. Is that 10 meters or is that 10 centimeter? We don't know. Somebody is just saying 2 centimeters above the sea floor. I have no clue. Is it 4,000 meter or is it just half a meter? So there is a lot of problems with doing the refinement and doing the curation. Not only in some cases they have a range, but you need to describe that in a format that we can actually search afterwards, because this is not searchable. You have no chance to get what you want if you are trying to search in a column like this. So we decided to, for example, with the depth, that we are using meter, and that's it. Not centimeter, not inches, and so on. So we have to go manually through everything and convert everything to meter. And the same for more or less all the 108 metadata fields. So we described all of the 108 metadata fields. We give examples of the range, for example, and units that we are using and everything. So it's well defined. In addition to that, we are using ontologies that is describing, for example, environments. So in total, we have 104, I think, different ontologies that we are using describing just the environment, the biome, the material, and the feature. I will come back to that and show you some examples. So everything has to be well described. So this table is actually 10 pages long, just describing each of the metadata fields. And of course, we have to convert it from a table format, human readable, to a machine readable. And this is also a way to, excuse me, to validate all of the fields. Because if it is an open space, it will more or less crash. So everything is controlled by using this tool that we developed. So we put everything into the Marine Metagenomics portal that is accessible for everybody. So you can go into it. You can look at the marine reference databases. Here is the link if you want to try it out, to look at the databases. So what we have currently is around 500 complete marine genomes. We have around 3,000 genomes in the MARDB, and at the moment we have around 1,500 samples from marine metagenomics. They are both contextual and sequence. They are manually curated, as I told you. 108 metadata fields. We have more than 20 links out to other databases if you want to have more information. And we are using ontologies and controlled vocabularies, because this is very important. If you want to search for something, you need to have ontologies implemented. So this is an example of the MAREF. If you go into it, you can actually look at where are all the samples. You can, if you want to click on one of those, you get more information about the sample or the genome, and you can actually click on that again, and you come into all the 108 metadata fields. So you can see all information that is available for that particular genome. And of course, what you see in blue is everything that is linked out to other databases. Another way to access the data is actually to go on the left side, and you will see a filter. So you can filter the project, the phylum, order, genus, and so on. In principle, we could have put up the 108 metadata field to filter, but that will take several pages actually. So we have just selected the most important ones. So what we can do is filter based on the project. We can filter based on the sampling environment. Phenotypical traits, collection date, depth, if you want to do that. Taxonomy, region or country. And if you are working with a pathogen, for example, you can select for the host, or if it's just associated with a specific host, you can search for that. If you want to search for a specific biome, you can select from this list. We have more or less 11 different biomes that we can select from. And if you just want to look at organism from marine mud, you can just select this, and you get only those bacteria that has been sampled from that site. So in this way we can get the genomes that we actually want to look at in one go, instead of trying to find those in other databases. The same with the environmental feature and the material. So if you just want to have some bacteria that is isolated from marine sand, you can actually just click on that and you get all of those. So this is important to filter out and to just get what you want. And you can combine all of these features if you want to do that. So this is just an overview of what we have in the database. If you, for example, go to ENA and search for marine, a complete genome, you get around 100. And most of those is actually from a marine center that has been sequenced human pathogens. So that is what you get out if you don't have a good curation of the data. So you see the difference between the complete ones and what is not complete. So here we have around 100, and we have around 200 up here families. So it's a huge difference between the genomes that has been completed and that is not completed yet. So if you want to search into these databases, it could be to have a look at both of these databases, and you can pull out whatever you want. So the sequence database is of course based on what is already in NCBI ENA, and that is annotated using this pipeline that has been developed by NCBI. All of the genomes in the RefSeq has been run through this pipeline. It's not the best one in the world, but it's still that is used by NCBI to predict the genes and annotate those. What we found out was that 20% was actually lacking in the database if you in ENA if you try to download it. So we had to predict and annotate those using the Proca software. The MARCAT, we are using MetaPipe to predict and annotate the metagenome samples. And we have looked at this before, so the difference between what Rob he was telling about is that we are using the assembly step because what we want in a gene catalog is full-length genes, not the short ones. So that is the major difference of these two pipelines, more or less. So the BLAST, you can, if you have a sequence, you can just put it in and run it through the different databases. You can download the contextual database if you want to have a look at all the metadata for each of the genomes. And you can also, of course, download the sequences from each of the genome and the metagenome samples. So it is not easy to make a database, so it is a lot of people that is involved in doing this. Of course, the manual curation takes a long time to go into the literature, find all the information and put it into the database. And also, so Inga Alexander is here, Esben and Eric is a part of this team that has developed this database. So you can find it at the MMP website, and you can contact us if you want to improve it. Please contact us and we can see what we can do. And that's all for the databases.